Good morning, good afternoon, or good night to everyone, those who are listening to my video right now. My name is Kirandeep Kaur, and today I'm going to give an explanation on a biological topic, Chemiosmotic Hypothesis. Chemiosmotic hypothesis has been put forward to explain the mechanism of how actually ATP is synthesized in the chloroplast. Okay, so to understand this, let's first of all draw thylakoid and see what all is present there so that we can understand the exact process of this ATP synthesis. So now let's begin. Okay. So let's first draw. So first of all, this is our outer thylakoid membrane. Let me write here. Outer thylakoid membrane. Thylakoid membrane. Now the next membrane we have is our inner membrane. And we call it as inner thylakoid membrane. Inner thylakoid membrane. Okay, so this is our inner thylakoid membrane. Okay, and this outside of both the membrane there is a space which is called stroma. So let's write here stroma. This space is our stroma. Okay, and here is present our water splitting complex, and this complex is basically present on the inner side of the inner thylakoid membrane. So this is our complex for splitting water. Okay, so this basically protolyzes the water. Okay, and now next is present our NADP reductase. NADP reductase, and our NADP reductase is present on the stroma side. Okay, and the space inside is our lumen. This space is called lumen, and this is our PS2, and this is our PS1. Okay, here is our reaction center, chlorophyll. Reaction center, chlorophyll. Okay, and on the upside they are present our primary electron acceptor. This is our primary electron acceptor. So this is our primary electron acceptor of PS2, and this is our reaction center chlorophyll and primary electron acceptor of PS1. Okay, so I written it just one time and I just pointing it with the arrow. Okay, so there are our three electron acceptors present here: one, two, and three. These are our three electron acceptors present. Let me write it down there. So these are our three electron acceptors. Okay. So these are our three electron acceptors. And this is our this one. This is our what? This is our cytochrome. Let me write here. This is our cytochrome. Okay. So let's see that there is a sun. Okay. There is a sun. Okay. And when sunlight falls on PS2, okay, especially on the reaction center of the chlorophyll, when sunlight falls on it, then our electrons get excited. Okay, and it will be taken by the primary electron acceptor. The primary electron acceptor accepts the electron here which has been excited, and these electrons are going to pass through the electron transport chain. So this is our electron transport chain, and the electrons are going to pass through the electron transport chain. So when the electron passes from higher level to lower level, it releases energy. So during this process, our cytochrome, this is our cytochrome accepts a pair of protons. So what our cytochrome will do? It will accept a pair of proton. Okay, so it accepts a pair of proton from the stroma side. So it accepts a pair of protons from the stroma side by using this energy and pumps it into the lumen. Okay, so it will pump the pair of protons into the lumen from the stroma by using the energy which it gains from transporting the electrons from higher concentration, from higher level to the lower level, from higher level to the lower level. Okay, so here what we get to know about our cytochrome is that our cytochrome is electron transporter transporter, and it is also our proton acceptor because it accepts the proton from the stroma and then it transferred it into the lumen, and it also transported the electron. So it is also an electron transporter and proton acceptor. Okay, so now here as we have our water splitting complex right there. Oh my god, let's see there. Here we have, this is our water splitting complex, we have it. Okay, so now H2O, this means our water splits here. Okay, so what happens, it splits the water into a pair of electron, a pair of proton, and it also releases oxygen. Okay, so now the oxygen produced here is going to be transferred into stroma. So this oxygen is going to be transferred into stroma. So here this oxygen has been transferred into the stroma from the lumen. Okay, and a pair of electron is going to be transferred to the reaction center chlorophyll of PS2 because the electrons are continuously being used here in the electron transport chain. So that's why our electron pair is being transferred to the reaction center. Okay, here is, now as we know that here we have a pair of protons which is produced by the splitting of water, and also a pair of protons have been transported from the stroma to the lumen by the help of the cytochrome. So here we have high concentration of proton in the lumen and low concentration in the stroma. Okay, so this is understood. So now let's move there. Okay, so now there are our two particles. One is our CF naught and the other one is our CF1 particle. Okay, so these particles, these are basically proteins, you can say, are present. Our CF1 acts as an ATPase. So this acts as our ATPase enzyme. So let me write here enzyme also. Okay, ATPase enzyme. Okay, so our CF0 and CF1 are proteins. Okay, here they are acting as a channel for the transfer of protons from the lumen to stroma, because here we have high concentration of protons in the lumen as compared to the stroma. So here these protons are going to be transferred from higher concentration to the lower concentration with the help of these two particles, which are our CF0 and CF1. Okay, So as protons move down the gradient, it also releases some energy. So when these protons, let's say this proton is going to be transferred here. Okay. So when the proton is going to be transferred, so I'm here drawing zigzag lines just to denote that energy is being released in this process. So now our CF naught helps in the facilitated diffusion of our protons, and the released energy is used by our CF1 particle to add an inorganic phosphate to the ADP. Okay, so what happens in this case? Our ATP is formed. Okay, so here our ATP is formed by the help of the energy which is released during the proton transfer. So here our ATP is synthesized, and after that protons come back into the stroma. So protons are back here into the stroma. Okay. The electron in the PS1 also get excited. Now let's go up. So here again our electrons. Now electrons have been transferred here. Okay. So now here again our electrons get excited. Okay. And now these electrons get excited by the sunlight. Okay. These also get excited by the sunlight. Okay. So now after they are excited, they are also accepted by our electron acceptor, primary electron acceptor. Okay, primary electron acceptor of our PS1, and then through the electron transport chain. Now here also we have an electron transport chain which transport the electrons. Okay, these electrons to our NADP reductase. Okay, so they got transferred to the complex NADP reductase. And now here what happens? Here our NADP reductase reduces the NADP by the help of the protons. Here what happens in this case? Our NADP reductase is going to be reduced. Okay. And now for this process we require protons, and these protons is going to be transferred here. So we get protons also here, and by the help of the NADP reductase our NADP got reduced. Okay, so now our whole process is complete here. Okay, so let us see the bigger view. You can even take its screenshot. Right now it's looking a bit messy, but I hope you will understand it very well. So this is the whole process. This is the whole mechanism of chemiosmotic hypothesis. I hope you all enjoyed it. Thank you so much. Have a nice day.